Hello, students. I want to use um this time to talk about um to talk about the glucose homeostasis. So just want to make it more complete. I figured that although I present this figure to you, but maybe I didn't make it clear enough. I received questions from students, so I want to make it clear with this video. So glucose homeostasis, the purpose is to maintain glucose level in our blood. It's because that our brain need glucose to produce ATP in order to function, and our brain don't store glucose. So we need to keep the blood a certain level of this glucose. So it's maintain that glucose homeostasis is important for humans' um, normal function, physiological function. So how do we maintain glucose homeostasis? So you need to go through this homeostatic loop. And so you will have a sensor to detect the blood glucose level. And then you have to have an action to correct that. If it's too low, you increase it. If it's too high, you decrease it. So let's look at this homeostatic loop. In the blood glucose, we we will have two different pathways to deal with either increased blood glucose level or decreased blood glucose level. So let's do increase first because this is more common. No, not here, not here, here. So increased stimulus right here, stimulus increased. So yeah, so let's do increase first. And so when your blood glucose level is high, usually when you eat after a meal, then your blood glucose will increase very quickly. Then something has to detect it. And that's something, the sensor to detect it in our body is beta cell. So this is pancreas, and the beta cell is the sensor. So if I ask you what's the sensor for to detect the high blood glucose level, that is beta cell. So beta cell detect it, then they will send signal into the integrator. In general situation, our body has an integrator in our brain, but here the integrator is beta cell itself. So it detected there are sensors in the beta cell, detect it, and just by beta cell itself, it respond to it, it directly respond to it. So the integrator will next send signal to the effector. So when signals sent from the effector, the integrator to the effector, we can go through a nervous system or we can go through hormonal system. So here you use hormone. This hormone is insulin. So it tells the effector to do something in order to reduce blood glucose level. And in our temperature, in our breathing, in our blood pressure, those we talk about in the lab, those use nerve, those use autonomic nervous system. You'll use nerve uh, to transmit signal from the integrator to the effector. Effector is a noun. So let me go here. So stimulus sensor is the organ, the anatomical organ to sense the stimulus. Integrator is a noun. Effector is a noun. So these two are the anatomical organ. So integrator will send signal to the effector. Response is an action. So these transmission from these anatomical organ to these anatomical organ, it usually goes through it either goes through the nerve or go through hormone. So it can release hormone to affect it, or it can send out nerve signal to tell the effector to do the work. So in this case, the integrator will send out signal, which is insulin, to tell the effector. When the blood glucose level high, the effectors the insulin will directly act on these effector. These effector include the liver, include the fat cells. It also has muscle cells. So insulin will act on these three to ask them to take in or uptake glucose from the blood, and in order to decrease blood glucose level. So that is the homeostatic loop when blood glucose level is high. So if we have these homeostatic loop, going to write about these loop, talking about the stimulus, that will be high glucose level. The sensor to detect high glucose level is beta cells. Beta cell has the sensor portion to detect the high blood glucose. Low blood glucose won't cause beta cell to do anything. Beta cells only sense these high blood glucose level. Then it will integrate by itself, integrate the information by itself. Then it will tell the effector. The way it tells the effector these arrow is either nerve or hormone chemicals. So in this case, the beta cell release insulin. Then insulin acts on effector, the anatomical organ. So there are three anatomical organ will be affected by insulin. We have the muscle cells, fat cells, and the liver cells. These cells has the capacity to store excessive energy in our body. So if you eat, you won't burn all this fuel at the same time or right away. You will store some of those. So when you are hungry, you can use these as your fuel to maintain body's function. So muscle cell can store these excessive glucose. Fat cell can convert this glucose to become fat and store in your fat cells. Liver cells can store these as glycogen. Most of the time it's glycogen. Sometimes it can store as lipid, but it's secondary option. So usually store it as a glycogen. And triglyceride, sorry, fat too. Yeah. Yes. No. Glycogen, that's more accurate. Yeah, glycogen. And then these cells will do the action. The muscle cells will convert the glucose to become glycogen. Liver cell will also store the glucose as a glycogen. Liver cell can also have some fat, but really the fatty liver could be it's kind of like a not the optimal option for liver to store the excessive fuel. Then the fat cell will convert the glucose to store them as glycogen. Oh, sorry, the triglyceride. Fat in the fat cells, and then by doing so, you decrease the blood glucose level. So this is the homeostatic loop for hyperglycemia, so high blood glucose situation. And then if the blood glucose level is low, so if it's low, then our body has another sensor to detect it. The sensor to detect the low blood glucose situation is alpha cells in the pancreas. And when alpha cell has a sensor portion, it will detect it. And then itself is also integrator, so it will respond to it. It will quickly release hormone to tell the effector to do something. Alpha cell will release glucagon. So glucagon is a hormone released from alpha cells. It will go through the blood, and then it will circulating. When it see the liver, it will tell the liver that, okay, I am a messenger. I was sent by alpha cells to tell you to release glucose. I know you store some glucose in you, and so now we need glucose. Please release some. So glucagon will tell the liver to release uh, glucose, and then glucose level increase, you return the glucose back to the normal level. So this is um, the other homeostatic loop. For regulating glucose, we have high glucose situation. We also have low glucose situation. So if we are going to fill out a homeostatic loop for hypoglycemia, the low glucose situation, then we go to this one. This is a slide that I didn't provide you earlier, but I will update this one on Canvas. So you have uh what's that? No, all right, let me correct it. All right, so if we are going to write about the homeostatic loop for to correct the low blood glucose level situation, and so we can write something like this. So here is that the stimulus will be decrease in blood glucose level. And so if it's low, then what gonna detect it? The thing gonna detect it is alpha cells in the pancreas. So this one, this organ system has the sensor to detect these glucose level. So sometimes you can think about glucose, you thought that that's a chemical. You may think that our body may have chemoreceptor to detect glucose level. Well, if we have the chemoreceptor, we certainly can detect glucose, right? You test glucose in your tongue, you can test it sweet. So you can test it. So we do have the chemoreceptor, but those for glucose, but those are not the one we used to maintain blood glucose level. Um, we use something that's more closely associated with the integrator itself. So this sensor is integrated, is part of the alpha cell. And so if the glucose level is low, this is critical because you need to maintain the glucose level. And so glucose, then alpha cell will respond, will detect it, integrate this information in the cell, and then it will tell the effector by releasing glucagon. So this arrow from the integrator to the effector, it could be a nerve, so it can be a wire, direct calling, saying that, it's just a wire that to send signal over, like nerve, or it can release chemical. So it release chemical. In this case, they release chemical. This chemical is glucagon. When they release chemical, release them into the blood, we call it hormone. So this is a hormone released into the blood, circulating around, and they don't see anything that they can bind to. And some of those they can see bind to and provide its function. Those are basically the effectors. The effectors are the one to get affected by the hormone. And so here is that for the glucose homeostatic loop, they will bind on the liver to tell the liver uh, to do something. Then this effector will have an action. This response is an action. So the effector will release glucose. And so we can increase glucose level in the blood and return glucose level back to normal. So that is the glucose homeostatic loop. I hope this make it more complete about this portion. All right, let me know if you have any questions, and I will see you next time.